Are we online now?
Yes, we are. Yep.
Yeah, okay. Greetings from OCP North America. My name is Amarjit Basra. I work as a chief scientist with OCP North America, and I will be your host for today's webinar. We are really grateful that Professor Erik SMOLDERS from Katholieke University Leuven in Belgium kindly accepted our invitation to give this very Africa-centric seminar, strategies to enhance phosphate fertilizer use in highly weathered soils of Africa. Welcome, Erik. Great to have you amongst us.
Hello.
So this is going to be a diverse group, people attending from different corners. So good morning, good afternoon, everyone, wherever you are. So we're very happy to have you here. So we'll start with the conversation with Erik, and then, as people pour in, then we'll start with the actual seminar. So before that, let me share with you the Agricultural Scientists for Africa group. Let me do that. Please bear with me. So let me briefly introduce you to the Agricultural Scientists for Africa group. So this group was created last year at the annual meetings of the American Society of Agronomy, Soil Science Society of America, and the Crop Science Society of America. So collectively, these three societies are called like Tri-Society. And there's a big membership, around 4,000 members attend every year annual meeting. This year it is going to be in Arizona, Phoenix in Arizona, but it has become a virtual meeting this year. So we started this group, being based in North America, but how to bridge the link between the scientists on the African continent and working anywhere else, whether in North America, Europe, or elsewhere, right? So that's how we have brought in also like Erik, who has been working on agricultural problems related to Africa. So the effort is that whether a scientist is of African origin, could be students or scientists working on African agricultural problems, or they could be working anywhere in the world related to a project related to African agriculture. So that's the group, and the need was felt within the Tri-Societies, being the international dimension of the Tri-Society, that how we can foster collaboration and bring people together. So the kickoff was done last year, as I said, and about more than 70 people joined in the kickoff meeting. And the opening remarks were given by Dr. Ratan Lal, who is this year's World Food Prize winner. And this year, in November 9th, we competed for a special symposium proposal, which the Tri-Society asked for proposals, and we put up a proposal on excellence in African agronomy, which was awarded by the Tri-Society. So some funding was given to us to conduct this. So it will be a virtual meeting, and so a number of eminent speakers will be, attending this special session, which will be cross-society, across the three societies, across the various divisions within the Tri-Society. So that event is mentioned here under Save the Date, the ASA Special Symposium on Excellence in African Agronomy. And you will identify some of the people, like I worked closely with Pedro Sanchez, who is another World Food Prize winner on this proposal. So he will be speaking, others will be speaking. So we can share with you the details of the speakers and their topics. But and so this webinar, so we'll be bringing you two webinars. So Professor Erik's is the first one in the series. So this is very related to Africa because we are a group dedicated to agricultural scientists for Africa. So the topic is also very relevant. And then we'll bring another speaker who will also speak on an Africa-related topic. I will project that after Erik's talk, the second webinar, that agricultural scientists for Africa will work work with UM6P, RUFORUM, and others to, and that webinar would be next week. So with that introduction, I will bring back the banners.
Okay.
And all right.
Yep.
Is looking good?
Yes, looking good. Yeah.
Yeah. Okay. So Erik, before we get starting, maybe in a couple of minutes, maybe could you tell us about your university? I know it is one of the oldest universities in Europe and also very highly ranked among the best universities for research and high quality education. So.
Yeah, indeed. We are a rather old university. Started 1425, but not with agricultural sciences. It's only in the last 130 years, I think. No, it started with theology. We are a Catholic university. There's not many left in Europe that are still termed Catholic universities. Having at the moment, let's say most disciplines, we are 40,000 students spread over different campuses in Flanders of Belgium, so the Flemish part of Belgium. Agricultural sciences is now called bioengineering because it's much broader than agriculture. Probably related to the fact that the research on agriculture within Flanders is very limited. We have mainly horticulture rather than broad scale agriculture. But of course, there's a lot of things on genetics, on biotechnology that we do in our faculty. We do have a long-standing tradition in research for development in terms of also tropical agriculture. We have a very large banana collection. I think that's the main collection of the biodiversity of bananas is hosted in Leuven. For reasons, it's just for sanitary reasons that we have that collection here in Leuven and not in Africa for diseases. And then I think our soil science group is quite well known. We have been active maybe 40, 50 years. One of our famous soil scientists is called Professor Dudal, who is known for soil classification. So the World Reference Base is now one of his, is a follow-up of what he did. He worked at FAO and at our faculty. And then followed by, some famous soil scientists like Professor Dekkers, Professor Ulmerks, who is also, who is part of that presentation today. I think we are very active in Ethiopia, in Tanzania, Madagascar, Congo, of course, because that's the former Belgian colony. Nigeria, members of our group have been active at IITA a long time. My colleague Ulmerks has also been sitting on the board of IITA until recently. So I think we have been quite active, and as long as I've been working now 30 years in the lab, and we always had African people, PhD students working in various themes and on soil fertility. So I think we have
great. And one of the purposes, this kind of interaction, UM6P is positioning itself to be, a center of excellence for related to agriculture, agrobiosciences, especially the School of Agriculture, Fertilizer, and Environmental Sciences. So it would be good to, for people to make linkages and see, to build also the collaboration. So I think with that, we can start with this webinar. And I have a distinct pleasure to introduce Professor Erik Smolders, who is with us today. from Katholieke University Leuven in Belgium. You heard a little bit about the university and its capabilities. So Professor Erik Smolders is an environmental chemist with specialization in soil science and environmental toxicology. He obtained his PhD also from the university, where he's currently a full professor. And he's teaching soil and aquatic chemistry and ecotoxicology. So you can see the environmental emphasis even in teaching. And you will come to know a part of his research, which is related to Africa, but his breadth and depth of topics is much beyond that. So previous to the university where he's currently a full professor, Erik has worked at Imperial College in London, also at the CSIRO in Adelaide, South Australia. He also worked in Austria, in Vienna, and also in US, University of Wisconsin-Madison. So a lot of good experience there. And I really want to thank Erik for accepting our invitation to be with us in a highly interactive webinar. So much of his research focuses on bioavailability of contaminants, heavy metals, and plant nutrients in soils as well as water. So his main expertise, one of which is also very relevant to OCP or UM6P, is cadmium and phosphate bioavailability in soil. So that's a topic, which is a focus area also at UM6P and very relevant to OCP at the industry level. And current projects in his lab focus on the fate and effects of phosphorus in soils and fresh water, also cadmium accumulation in cocoa, phosphate organic matter interaction in soils, especially in the context of Africa, and that's the topic of today's webinar. And also the environmental risk assessment and effects of metals on plants. So remarkable productivity of the group. His group has published about 290 peer-reviewed publications and contributed to 8 books. It's amazing productivity, and I also see that these publications are highly cited. If you look at his H index, or we say that how much significant impact they have made. So with those words, I will invite Erik to present his webinar, and I will project his slides, and we'll go from there. So I will remove myself in the background.
Yeah. Can you just confirm that you can see my screen okay, Amarjit?
Let me remove those ones now. And brand. I can see Erik, yes.
Yes, and the pointer. Yep.
Yep.
Okay. Good. Then I'm all set, and I'm ready to go for another, say, 45 minutes. I think I will go give that talk. So, ladies and gentlemen, thanks for attending this seminar. I'm going to talk about strategies to enhance phosphorus fertilizer use in highly weathered soils of Africa. So, to increase food productivity in Africa, we need to capitalize the soil fertility. There is among the 13 or 14 different mineral elements, there is mainly phosphorus as a limiting factor. Of course, nitrogen is also quite limiting, and there's regions where you have zinc and boron deficiency, sulfur deficiency we have identified. But the one that I'm going to talk about is phosphorus, and that is the one that is most often limiting in highly weathered soils. I'm giving this talk on behalf of other people of our group. I'm indebted a lot to Roel Merks, who is not attending today, but he has been leading a lot of that research in our group. I've been in the past following it up a bit, and then in well, let's say since 10 years, I'm also really active in that research myself. And two other people who were mentioned here, two persons as Pieter-Jan de Bouw and Mieke Verbeek, two postdocs from our group, have contributed a lot, not only the slides, of course, but into the research. And I will mention some of the work that they have done, and also I will mention work from other people. I guess you all have been looking to the picture in the back. Let me go to that picture. It is just an illustration how strong that phosphor deficiency can be. It is Madagascar, the highlands of Madagascar, that have highly weathered soils. Many of them are not used. The upland areas. In the lowland, there is paddy rice. You don't see that, but in the upland, there is no increasing attention to also cultivate these areas. When you remove the grass and start to cultivate, for example, the upland rice or soybeans or whatever, you often face limiting factors, and you think it's water, but if you irrigate, you still don't see many crops. And look, we have been now active since 2009 in Madagascar, and this is now what we call the iron mine. This is a field location where the effect of phosphor is the most pronounced ever seen. For 3 years, in the last 3 years, we have been growing rice and different classic fertilizer trials. And in the front right, you see the plots that are NK fertilized and irrigated, but we don't give P. And all these 3 years, we had absolute crop failure, whereas in the ones in which we fertilized with phosphor and irrigated, we had higher growth, and depending on genotype and treatment, we went up to a massive increase to 6 tons of rice grain, which is maybe the yield potential for that area that is rather dry and cold periods, etc. But 6 tons versus 0. So as we say in the lab, if you want to draw a dose response curve with a 0 intercept, you have to go to that place. In the previous field, not too far from that, we had shown that, well, it's in fact not a surprising result, but we had shown that when we combined phosphor fertilizer, so repeated triple phosphate application in these soils, that we could enhance the grain yield, and we had low and poorly responsive yields. We could go up to 5 or 6 tons in some years, and the highest yields what we found were when we interacted the TSP with farmyard manure. So when we combined farmyard manure with TSP, and these are the results. And I'm not giving the yields, but I'm giving here for the 3 years. I'm giving the phosphor uptake in the above ground matter, so grain and straw. And if you look at the third year, look first to the x-axis. Guess that most of you will be familiar with typical doses of phosphor in a field that you go for, well, depending on the economy, you go to 10, 20 kilograms of P, not P2O5, but P. In the presentation, everything is in phosphor units. So we give here up to 80 kilograms 3 years in a row in the same plots. Yeah, we do see finally a response, but the response is still not massive. And when we calculate how much phosphor we get for the phosphor we added, then you see what we call the phosphor use efficiency. And this is the central theme in my talk today. I will use that term, phosphor use efficiency, just to say, make the red line through the whole talk. Here, the phosphor use efficiency defined as the phosphor uptake in the above ground matter or in the grain divided by the applied phosphor. We have only a phosphor use efficiency of 10%, not more. So 90% of the phosphor is given to the soil and is, well, to capitalize, but we have a very limited efficiency. If we combine that with farmyard manure, we will increase that efficiency, and I will come back to that later. But if we want to increase or capitalize the soil fertility in Africa, we need a good return on investment. You have to convince the farmers that if they invest in phosphor, that they will have some benefit, and a central idea for that is the phosphor use efficiency. So we have to do whatever way by selection of genotype or better soil management or water management or what have you, increase the phosphor use efficiency. So it's just an index that economists may like to use, that phosphor use efficiency. And to give you a bit of background on where we come from, like say in Europe, where do we come from in the development of our agriculture? Well, our agriculture has been intensified in the last 150 years in Europe. But if you look at the last 55 years and starting in 1965, we had mean European phosphor inputs of about between 20 and 25 kg of phosphor per hectare per year. The phosphor uptake was about 4 kg of phosphor per hectare per year. So we had also a very low efficiency at the start. But as agriculture developed and the soil fertility rose, and also the selection of the suitable cultivars and interaction with nitrogen, in Europe, we went up to about 30%. And then we started to identify that phosphor was no more limiting and we could lower the phosphor inputs. And around 2007, we only applied, let's say, on average, 15 kg, and we had a phosphor output of 10 kg. So we are close to 100%. Well, we are at 70%. Of course, we can go lower. 2050 projections that was done by Satari and Pina's paper, that maybe then we reach a steady state so that we add to the soil the same amount that we remove annually from the soil, and at which we have 100% efficiency. Of course, economically most sustainable, let's say. Now, in Africa, the soils are so poor that the phosphor use efficiency extremely low. An example, we talked last week with my colleague Roel Max, where did you show phosphor use efficiency? And he recalled examples that they did in maybe 20 years ago in Vietnam, not in Africa, but in Vietnam, with similar soils, highly weathered soils, in which the phosphorus was really fixing. And look at the response curve they had with maize. They added a massive dose of up to 200 kg phosphor per hectare, which is of course not economically viable. That's far too expensive. But they wanted to identify the plateau. And what they found is that even the 200 kg was not yet a plateau. You see, it's still increasing the maize yields, grain yields for maize. When they repeated on the same plot the year after, they had higher yields, but maybe not yet a plateau. So they had then already applied 400 kgs. And then the year after, they stopped in the 200 plot and they stopped that, but they continued the 100 kg plots. The phosphor use efficiency was less than 6%, and the doses were still not optimal. So then you can wonder, why is that? And what does Africa have to do? Now, just for the context of Africa, in most soils in Africa, even if phosphor is a limiting factor, there is a negative phosphor balance. And already since the 1970s, there is more phosphor taken off by the crops than added, which means that the phosphor deficiency is increasing and increasing, the phosphor deficiency, with a few exceptions such as Kenya and Ethiopia, where the country averages show that there is a positive balance. Yeah? Also, you see that in these areas that the plant uptake is increasing, getting to a few kgs of phosphor, but still not at the 10 or 15 kg in which you have to go when you have no more phosphor deficiency. The phosphor fertilizer use is extremely low in Africa, and only in these countries like Kenya and Ethiopia, and also a little bit in Nigeria, the phosphorus are increasing a little bit. But if you look at the average mineral phosphor use in Africa, we still talk about very, very little numbers, and there's not a big change. If you look at these numbers here, so 1.9 2002, going to 2.5 in 2012, and then decreasing a little bit, and then maybe up and down, but definitely only a few kgs of phosphor. Now, why is phosphor so limiting in these soils? Well, let me take you back to the classic textbook of soil science about soil mineralogy and soil weathering. If you take in a pristine young soil, a soil that develops, in the x-axis is the development time, the weathering time. In a very young soil, there is nitrogen as a limiting factor. You have a pioneering vegetation with nitrogen fixing plants, and they will increase the nitrogen. But at the same time, as the soils age and weather, the phosphor is not only leached, but the minerals are changing. So you start from the feldspars and the micas going over the smectites, and then when they weather, they will transform into iron and aluminium oxides that trap the phosphate. And the phosphor will decrease in availability. And that's the classic thing that in young soils, nitrogen is limiting. Then there is a phase where both are limiting, but in older soils, the phosphor limitation starts to develop. And at the same time, iron and aluminium oxyhydroxides increase. And the iron oxyhydroxides that you can tell from the color often, the hematite that gives the red color. And then when you have a lot of iron, you see the red color, you're having phosphor deficiency. The total phosphor in these soils is often high, but it's not accessible, it's not available, which is, of course, the frustration of many people. There's a lot of phosphor in the soil, but it's not accessible. So we have to increase the available pool in the soil. The only chemical slide, no, there's 2 slides of chemistry. I'm a chemist, and I love chemistry, but I will limit myself to here. Why is phosphorus so much limiting when there is a lot of iron in the soil? These are 2 neighboring iron octahedrons. So inside this octahedra is an iron atom. They're surrounded by 6 oxygen atoms. And when there's 2 neighboring octahedra, then phosphate, this is the phosphor atom in yellow, and the phosphate ion, the phosphor can form what we call an inner sphere binding on 2 neighboring, we call it corner sharing octahedra. It forms a bidentate, it's strongly sorbed, and there's very little phosphor in solution. So very simple, as iron and aluminium oxyhydroxides increase in soil, when they become dominating, they will trap the phosphor and keep it out of solution, and the plants cannot access it anymore. When you take such a soil and you add phosphor to that soil in the lab, you mix the soil with a phosphor salt and you measure the tiny little bits what is in solution, then you get the classic sorption isotherms. Well, typically a sorption isotherm, you put the solution in the x-axis and the adsorbed in the y-axis, but now we do it as from an agronomical point of view. You increase the phosphor to the soil, and then you see the phosphor in solution increasing. First straight line, and then suddenly it breaks through and you have the steep increase. It depends on the soil. The black dots are soils with very little iron. The black triangles are a soils with extreme amount of iron. The term phosphor saturation index is the phosphor dose that is required to yield about 200 microgram phosphor per liter. 200 microgram per liter is here in that extract of the soil. So the soluble P, 200 microgram per liter, is about what plants need for full development. So when phosphorus is absolutely not limiting, then the phosphorus solution is typically around 200 microgram per liter. If we have this soil here with the open symbols, then the phosphor saturation index is 600. We need to add 600 milligrams of phosphor per kg of soil to reach this value. So that's the amount of phosphor fertilizer expressed in milligrams per kilogram that you need to add. To give you an idea of the importance of iron oxides, I take back all the data that we have published, in which we collected a wide range of soils from highly weathered soils, most of them from Africa, with a few from Vietnam as well. They're all acid. So you see the pH here, 4.6, even one that was 3.9, one from Kenya with 5.3, but they're all, that's typical. They're highly weathered. They have lots of iron and aluminium, and they're acid. We rank them here in this table from low to high iron. And here is the measured phosphate saturation index. When the iron is low, the PSI is low. This soil of Madagascar, which is an undersoil from close to a volcano, has extreme high iron concentration and has an extreme phosphorus saturation index. You see, when iron goes up, the PSI goes up as well. There's a few things like here in the soil of Kenya, the pH is higher, it has high iron, the PSI is a bit lower. But that is because the pH is another factor that determines the PSI. As a rule of thumb, acid soils, so low pH, have a higher PSI as well. So it's not only iron, but also the pH that matters. Now, what does that mean for our phosphorus use efficiency? Let's take the very first soil, Deso, with a very low PSI, 15 milligrams per kilogram. If we want to have optimal phosphorus doses to the soil, optimal for plant growth, we need to add at least 15 milligrams per kg of soil. What does that mean in terms of dose to the field? Well, if we take this value, about roughly multiply by 2, we end up the unit kg per hectare. For those who like the maths, it's depending on the bulk density, of course, the depth of penetration in the soil. So if we incorporate it in the first 15 centimeters, and then you have the hectares, then you end up with this value multiplied by 2 is 30 kilograms. So 30 kilograms is the dose you need for Kenya, but the dose for here, that soil in Madagascar, is more than 2,000 kilograms, so more than 2 tons of phosphorus. Now, at optimal P dose, and if you have a normal, I'll say a normal yield potential of about 7.5 tons of grain when nothing is limiting, then you have a typical phosphorus uptake of 15 kg. So for this soil, the phosphorus use efficiency would be still quite good. You add 30 and you get 15 in return, so your phosphorus use efficiency will be 50%. Now, this phosphorus use efficiency is predicted to be high for that soil in Kenya, but if you would start to capitalize the soil fertility in these soils of Madagascar, you will end up with a phosphorus use efficiency of only 1%. So your return on investment will be extremely low, and nobody will want to start in such areas to capitalize the soil fertility because of the very low return on investment. To give you an idea about how good or how bad that just very simple chemical test is, let's now go back to that soil of Vietnam. That was the Dak Nau soil, which has a PSI of 290. So you need about well, this value times 2 gives you about 600 kg of phosphor that you need for optimal phosphor supply. This is the slide again from that old study in Vietnam. We added 200 and it was still not enough. The year after, we added another 200. So we had then 400 added in total, and we had reasonably good yields. And the year after, we stopped. So these open points are the ones in which we did not add any further. So in total, the cumulative dose was 400. And what we predict by our chemical test is that it needs at least 600 for optimal. So we are not too far from that, but the reality is that gives you a very low phosphor use efficiency. And it's not finished. There is another thing which is well known in soil science already since 1950s, and that's the aging of phosphor. You can add that phosphor, you can add, say, 200 kg, use 10% only, and the next year you think you have 190 left. But what that 190 left has a residual value that is lower than the fresh addition. That's what we call the aging effect. And in a chemist way, this is what the how chemists looked at. So if you add phosphor, the soluble P is increasing for fresh phosphor addition. If you leave it to age 1 year later, the curve looks like that. The reason for that is that the iron oxides are microporous. So a lot of these iron oxides have outer surfaces and inside surfaces. When you give a fresh dose, you only look at absorption at the outside. But if you give sufficient time, phosphor will penetrate into the pores and the solubility will be even further lowered, because then the inside pores and the inside binding sites will also be used. Another way of plotting this, the soluble P over time gives you this curve. If you have soils with low oxalate extractable iron, so that's the amorphous iron. If it's rather limited, it's this. If you have high iron, you have immediately a strong sorption, but also the aging keeps on going. 3 years ago, we did a systematic study of phosphor fixation, but what we did is we did not wait 1 year or 2 years. We did not have the time for that. It was done with a PhD study in which we did 20 days of um, adsorption time, but of course 20 days of adsorption with agitated suspensions. That's of course overestimating the speed of reaction in the field where you have these aggregates. In the lab, you can accelerate that by just agitating. And you see here typical cases, a low iron solution, a high iron solution. The time here is on the log scale on the x-axis here, 2 minutes up to 20 days, and you see it reacts in high iron soil. Immediately you drop to low P in solution, so that's the phosphor in solution, immediately to low, and it's going on. So the fixation does not stop. And when we combine everything of all different soils, highly wetted soils, not and young soils, and soils from the temperate region, what we had is what we call the aging factor, that the factor change in soluble P between 1 and 20 days in contrasting soils vary between 1.1 to 6.9. With 6.9, imagine what that gives in the field. That's the factor reduced bioavailability as well. So that's all the phosphor fertilizer that decays away, let's say that is going to be fixed and is not reduced, is not available anymore. If we plot that factor, we call that the aging factor, to soil properties, then in fact, the most important factor that comes out is the ratio of oxalate extractable iron to the total iron, well, the DCB iron, dithionite citrate bicarbonate, that's more or less the total iron in soils. And that tells you that if the oxalate is the amorphous fraction, if the amorphous fraction of the iron is higher, you have more aging. Amorphous fraction is iron that is microporous. So you can have a lot of iron, but if it's really crystalline, then it has very little internal pores, and then the phosphor can react, but then it will react immediately and then it stops. If you have a lot of amorphous iron, then it will continue to react. Before we give you a list of possible options to improve phosphor use efficiency, I give you a few slides on diagnosing soil phosphor deficiency. In fact, what we want is, do we know how to test soil for phosphor deficiency? Is that very simple or is that complicated? Well, for those not familiar with the phosphor test for soils, let me tell you this. There is different soil tests that have been developed in the soil science literature and also in commercial labs. And it's enormously dependent on region where you are, which soil test is used. The classic, the most widely used tests for phosphor are the Olsen test, developed in the States, US, also used in, for example, England. But then there is the Bray test, also used, developed in US. There is the Mehlich test. There is an oxalate extraction. There is a calcium chloride extraction, ammonium lactate extraction, and then in more recent literature, the DGT test, so something with a diffusible phosphor and so on. And there is, say, a lot of controversy. Which test is now best? Well, in fact, the general rule is use whatever test you want as long as it is calibrated. And for Africa, what we noticed is that there is very little calibration done. Calibration done with field trial responses. I mean, how do you calibrate a test? You take the source before you fertilize, you analyze the phosphor in with a given test, and you have to calibrate that to fertilizer response trials. And typically what you put in the y-axis is what we call the relative yield. So the yield relative to the dose at which you have no more effect of the phosphor. So when you're and then you can compare also different soil tests. We have done that recently for European soils, and we took samples of the most important long-term trials of phosphor where people have been adding phosphor at different doses for repeated years up to more than 100 years to identify response curves. And we took samples from the most important long-term trials in Europe and identified which one was the best. And what comes out for the European soils is that the Olsen and the ammonium lactate test, that they are most, well, give the best responses to the relative yields. So you see in the y-axis relative yields and the x-axis the phosphor test. This is the oxalate extraction, this is the ammonium lactate extraction, and then you have the other test. In terms of R square, here you see that Olsen was the best one, closely followed by ammonium lactate. Other tests such as DGT test here in the bottom left, which is a test for measuring the fluxes of phosphor from the soil towards a zero sink, so something that adsorbs phosphate, was not bad, but was definitely not the best among these 5 tests. Earlier work in our group done on highly weathered soils, such as including many soils from Africa, showed that in general, the DGT test, which is here on the bottom right, was outperforming the other tests, such as the Olsen test, by far. Look here, the data for maize, relative yield versus the test here in the x-axis, the black full points, or the results for maize, was definitely much better for DGT compared to the Olsen, which only gave a lower R square of 0.52. Also, there's other ways of looking at in terms of discriminating deficient from non-deficient soils. In that soil, for that type of soils, we concluded that tests such as the DGT are better suited. We've also been able to explain why in some soils, things like Olsen tests are better or ammonium lactate extraction tests are better. And in other soils, like tropical soils, why tests such as calcium chloride or DGT are better. And I take you back to the old story of quantity intensity relationships of solutes or nutrients in soils. What do I mean with that? The intensity is the concentration of the nutrient in the soil solution. The quantity is the total accessible quantity of the nutrient in the soil in the solution plus the quantity that is adsorbed on the soil solids and that can replenish the soil solution when the plants remove it. So that is the buffering, the buffer of nutrients. Now, we put all the data that we had from European source and highly weathered source in one database and we plotted the relative yields. And what we found is there's 2 market regions. In the colored lines are the equal relative yields. So x-axis is Olsen, that's a quantity. Y-axis is soluble P, that's calcium chloride. Intensity in the y-axis, quantity in the x-axis. And we plotted the ISO concentration, so the lines in which we have equal relative yield. We call that the isoballs, at which relative yield is equal. So for example, here quite optimal, 95% relative yield. So all the blue lines, the points on these lines combine all the points that have a relative yield of 95%. When we have very little soluble P, when there is an enormous sorption in soils, such as the highly weathered soils of Africa, we are in this zone here, the lower range of the solubility. In this range, it is clear that to go from relative yield of 60 to 80 to 95, it is clear that you have to do it by increasing the intensity. That means increase the soluble piece. However, if you're in the zone where the soluble P is extremely high, then there is, like say this line, there is no point of increasing the solubility anymore. It's saturating. However, you can be in that range here where you can increase the solubility. It doesn't increase the relative yield, but in this range of high solubility, it's a matter of increasing the total quantity. So if you have soils with very little iron, for example, sandy soils, which have very little phosphor binding power, and you want to increase the available piece in the soil, so increase the relative yield, you need to increase the total quantity. So you need to increase the Olsen P. The soils from the temperate regions are most often located in this range, whereas tropical soils are located in this range. That also shows why tests such as Olsen are not really the best choice for tropical soils, whereas that Olsen test is better for the temperate soils. And maybe that's also the reason why in the US and in the UK, in the relatively younger soils, this test has been developed and used. For tropical soils, we need a test that is extremely indicating the solubility. It also means in our adventure for looking for higher phosphor use efficiency that we mainly have to look at all strategies of managing the source and managing the fertilizer protocols to increase the solubility, not the quantity, but increasing the solubility. Right? That brings me now to the point how to enhance the phosphor use efficiency and overcoming phosphor deficiency. The first thing I'm going to talk about is about our studies on the interaction between organic matter and phosphate. What is given here on the slide is an iron oxide. It is goethite structure. You don't see the phosphor bound to it. The phosphor, so when you have a phosphor ion, it binds to this iron octahedra. But what is in the blue shade is an organic anion. Here it is citrate. Of course, citrate is not really the molecule that is most prevalent in soils, but humic substances they do have carboxylates as their reactive sites, and these carboxylate can bind to the iron oxides. Carboxylates are anions that can form an inner sphere binding or just an electrostatic attraction with a positively charged iron oxides. And by doing that, they block phosphate binding sites. To make it really simple, increasing organic anions in soils, such as the humates or here the citrate as a model, block phosphor binding sites and can keep phosphate more soluble. One of the more recent works that we have been doing is studying the organic matter phosphor interactions with the idea of what is the effect of organic matter on the phosphor desorption. So we were studying soils in which we desorbed phosphor from soils. So we took soils as they are from different locations, from long-term trials with phosphor, long-term trials with phosphor, and a wide range of soils from a bit everywhere. We did desorption test and identified what is the role now of organic matter. And here are the results. These are very recent results of phosphor desorption tests that have been performed even during the lockdown. We have been working in the lab to continue that test and not stop that test for testing phosphor desorption for over 160 days. And this is now a phosphor desorption, how much phosphor can be desorbed from the soil as a function of time for, in this case now, two soils, one with a high carbon to iron ratio and one with a low carbon to iron ratio. Of course, not surprising, when you have more organic matter in the soils, you desorb more and, not unimportant, faster. This one also desorbs quite some phosphor, but it is slower. We describe these desorption curves with, say, a two-pool model, with a fast pool that comes out in a relatively short term, followed by the slow pool that desorbs it more slowly. The fast pool is most relevant for bioavailability. When we took all the soils together, I only gave you here now two, but we had a lot of soils that in our collection on which we did the phosphor desorption terms. What comes out is that the labile fraction, so the fast pool as a fraction of the oxalate extractable P, ranged between 5 and 25%, that there's two groups. Soils with high fertility, with a high phosphor to iron ratio, where the phosphor is, say, more loosely bound, there is a high fraction. But in the soils with a low phosphorus status, and of course, these are definitely all most of the soils from Africa, where the phosphor to iron ratio on a molar term is much less than 0.3, but we took that threshold. In that range, we found a surprisingly strong correlation between the labile fraction and the organic carbon to mineral iron ratio. So the ratio of carbon normalized per iron. What does that mean? We all know that soils in Africa, in highly weathered soils, are rather low in organic matter for obvious reasons, because it's warm and humid, organic matter decays away fast. If we can ensure that there is a reasonable amount of carbon per unit iron, then we can ensure that the phosphor is more soluble, that it is less fixed, and that it can be more desorbed. This is one of the factors that explains our interaction in the field between adding farmyard manure together with phosphor. Again, is that slide from Madagascar in which we studied the interaction between farmyard manure and phosphor. Remember, I showed you that if we add just mineral fertilizer in that soil without organic matter, we have a phosphor use efficiency of about 10%. However, if we combine that with farmyard manure, you see that the slope of that line is definitely steeper, and the phosphor here in the x-axis is including the phosphor that we added with the farmyard manure. Of course, farmyard manure is also a source of phosphor, but that is taken into account. And you see that even after correcting for all the phosphor, the blue line gives much higher response. Note that in all these plots, all the plots that I'm showing here, and also the plots that I will show below, nitrogen and potassium, and also in some conditions, micronutrients, they're all dosed in all the treatments. So what we look at is only the phosphor response. But what comes out of this study is after 3 years, that the combination of farmyard manure with phosphor gives you an enhanced phosphor availability, which is partially related to the fact that the organic matter can keep the phosphor more soluble. But it's not the only reason. So we repeated, so that here in the last 3 years, we repeated it once more, and we included also genotypes. So we worked together with Matthias Wisua also for selecting suitable genotypes of upland rice, so that are more suitable for low phosphor tolerance. But what we measure in what we identified in that region is that there is something else than only keeping the phosphorus soluble. That is the effect of farmyard manure on the pH. That, well, also that is not extremely new, but now we did the study in interaction with the TSP and in interaction with genotypes, but we found that the pH effect is large. When you add farmyard manure to an acid soil, the pH goes up. This is in blue the control pH and the y-axis, and the x-axis the day after the application. And when you look at the immediate effect of adding a lot of organic matter, you see an enormous effect on the pH. An increasing farmyard manure application, increasing pH, is of course very good for the plants because of the aluminium toxicity. Plants do not like acid soils, aluminium toxicity, manganese toxicity, also quite prevalent when you have, and definitely when you have a lot of water. But these things have to be avoided. By adding organic matter on the short term, you largely increase the pH. After a while, of course, you have some, again, acidification due to the nitrification and the nitrate leaching. But if that is not too much, then you can avoid that effect. What I have not yet told you is that in all the tests that we have been doing that with that interaction organic matter and phosphor is that we have been co-locating organic matter with phosphor in the planting hole. If you want to save on your soil amendments, then ensure that you add everything locally. That work on organic matter application in planting holes is also not extremely new. I mean, that has been shown in the past and with all different plants, with coffee in Vietnam, I think. And we have been working a lot on the interactions of organic matter, what is behind, but definitely in this soil of Madagascar, the pH effect was one of the major factors. And when we did this here after 3 years, we found this here. Remember, this is the extreme soil within the control, no yield. 3 years, nothing. Adding organic matter gives you a reasonable yield of between 1 and 2 tons. Adding phosphor gives you about 2 tons. And adding the lot together is not 1 plus 1 is 2, but sometimes 1 plus 1 is 3. You see, for some genotypes, because each of the different bars is a different rice genotype, we can get to a great yield of 6 tons of grain, whereas in the control is 0. This is the phosphor that we added. Note here that we did not add these huge quantities. We did not go to these hundreds of kilograms of phosphor, despite the fact that the phosphorus saturation index of that soil would indicate that you need definitely a few hundred kg of phosphor per hectare. But by adding it locally and in interaction with farm manure at reasonable doses in the planting hole, then you get this, well, reasonable doses in terms of dose per hectare, but locally these doses, 10 centimeter depth, are quite high. So you add quite a bit of organic matter around the planting hole of the rice seedling. In terms of phosphorus use efficiency, well, what you get is about 10% when you add just TSP. And when you do that with interaction with farming manure, you get to 14%. Again, that interaction. So we also, the statistics, that paper is under revision now, but the statistics are clear. Phosphorus use efficiency is significantly affected by the interaction with farming manure. What we also have is an effect of the genotypes in, well, depending on the year, there is definitely an effect of the genotypes. What we found is that the Fofifa and the Chomrongdangs, these were the ones that are the most suitable for the lower temperature than Madagascar, that they were superior. But we also did a liming trial. We included a liming trial to identify which of the varieties are more tolerant and which are less tolerant to the low pH. And there's, again, well, there is definitely also a genotypic effect. What this slide shows is that when you add nothing to the soil, just NK and water, that you have no yield, but when you add a bit of lime to the soil, that you do start to see effects, even if we don't add phosphorus. So the low pH is definitely another reason why we don't have yield. But if you add then lime and TSP, you get in the, well, reasonable phosphor use efficiencies that are similar to farmyard manure. So the story of farmyard manure is a combination here of a liming effect and an effect of keeping phosphor soluble by the organic matter phosphate interaction. Next topic, placement, fertilizer placement. We have been doing a bit of more research, and this is the work of that Pieter Jan de Bouw did on phosphor placement. Phosphor placement relative to phosphor broadcast, well, that is well known that you need to add the phosphor where the plant is, for the simple reason that phosphor is diffusing so little, only a few millimeters in a year, well, or millimeter or centimeter in a year in these soils. So you need to add the phosphor where the plant is. And that is, of course, well established that you increase the phosphor use efficiency, but there is more than that. Let me bring you to this cartoon. When you add phosphor to the left as a placement, you have a local fertilizer granule, and of course, locally, you have a lot of soluble P. When you do a broadcast, yeah, you add it to the top, and then yeah, it's a bit more this diffuse, it's broadcasted, and then you have this result. Now, broadcasting versus placement is not only, well, the placement is not only beneficial because you place it where the plant roots are, but there is something else, and that is related to the non-linear sorption. Remember these adsorption isotherms. When you locally add phosphor, you bring a lot of phosphor in solution, but you also saturate all the binding sites. So you make a local hotspot, and then you saturate the binding sites, so you immediately go to much higher concentrations than that critical concentration of 200 micrograms of phosphor per liter. When you take soluble P and you mix it into your entire soil, the same dose can give you very little only increase of phosphor because you distribute all your phosphor over all the binding sites in the soils. This has been shown by us and the group, and also in Adelaide by Finn de Vries, has been shown that, in fact, for these acid soils, weathered soils, you need to add phosphor locally, and you have to make a hotspot. In contrast with calcareous soils, you have to do it differently. You have to avoid phosphor precipitation as calcium phosphate, and therefore it's better in these soils to distribute the phosphor, so to avoid local oversaturation. But for the acid soils, it's clear that you have to place the phosphor, and that gives you much higher phosphor use efficiencies. Okay, back to the field. Stop chemistry. This is Tanzania, small plots for lowland rice, in which Pieter Jan de Bouw did his experiments on placement in interaction with genotypes in interaction with water. I'm only talking here with not about the genotypes, but the interaction of placement with water management. Definitely a phosphor deficient soil, so it had everything except phosphor. So it had nitrogen, it had urea, it had micronutrients, it had potassium, etc. Here is the adequate broadcasted P. Here is the micro placement. You could say, well, let's go for the broadcast, but until you see the economics or the doses added. This is a grain yield. In the x-axis, the 4 treatments. No P, a microdose of phosphor of only 3.5 kg, then a micro higher dose, and then the optimal broadcasting. Yeah, 25K. Look what happens with only 3.5 kg of P. You have already a nice increase of grain yield. And then we have these 3 water regimes. Field capacity, so you keep the soils moist but never saturated. So the water table is 20 centimeter below the surface. Wetting and drying, so where you periodically flood it and then you keep it always flooded, the lowland rice. Surprisingly, the field capacity yielded more at the microplacement compared to the optimal, where you added a high phosphor dose in that soil. This is a bit counterintuitive. People who've been working with phosphor, they always think that phosphor needs water for diffusing, because phosphor gets to the plants by diffusion, and diffusion is extremely sensitive to soil moisture. If you have air-filled pores, it cannot diffuse. So you need to add sufficient water. Now, well, field capacity is quite some water, It's not dry, it's not a drying stress, but it's not like water saturation. So why is that? Now, if you first look at the plot of the phosphor deficiency, oh, my slide do not move forward. There's a technical problem here. Oops. Don't know why that is. I have to go out of my slides. Oh, there is. Let me have a look here. Yeah, here. Okay. Here is the phosphor use efficiency of this treatment: 0, 3.5, 7 kg as micro P, and then the plus P of 25. If we look at the phosphor uptake and we calculate the phosphor use efficiency, we have a massive phosphor use efficiency under the field capacity at the lowest dose of 55%. That gives you definitely a return on investment in that situation. When you have a soil that is more wet or completely wet, that phosphor use efficiency immediately drops. So there is a significant water times phosphor interaction on the phosphor use efficiency. Much less pronounced at the optimal, but at the lower phosphor doses under phosphor placement, there's a large effect of the water treatment. Why is that? Well, it's related to the effect of the water on the roots. Let me explain you this. In the y-axis is the root efficiency, that is the phosphor uptake per nodal root. In the x-axis is the nodal root thickness score, between 4 on the right, the thickest roots, more than a 1 millimeter diameter, to the finer roots. So when you have finer roots, you have a higher root uptake efficiency. Not surprising, because the root surface area determines how much the phosphor can get in. I mean, under phosphor deficiency, its diffusion is limiting. So the larger the root interception area per plant, the better. So that is in itself not surprising that you find such an inverse relationship, that when you have smaller roots, then the root efficiency increases. But here are the treatments. For example, the blue lines are all the low phosphor treatments. At reduced water supply, so drier soil, you go from flooded soil submergence to water stress, more water stress, then you make finer roots, the reaction of the plant to the limited water. At the same time, that overcomes phosphor deficiency. And it's definitely true when you have micro phosphor placement, when you have that micro dosing. So also in the middle phosphor, you see that trend. So that is, root architecture was one factor explaining the larger root P uptake efficiency at reduced water supply. But there is another one, the phosphor diffusion from the fertilizer. Remember this scheme here. When you have soil submergence, the phosphor can diffuse further away and will get trapped. We modeled the phosphor diffusion under submergence and also under drier soils. And the result was that if you have submergence, you have more phosphor fixation. So you have less of that hot spot. Again, that is a small recap. If you want to increase phosphor use efficiency, you need increased solubility, and you can achieve that by making these hotspots. So hotspots are important in the whole process. Yeah. Okay. Next. We also did some modeling of the role of root architecture on phosphor uptake. We did that for various treatments, low, medium, high phosphor, and then at field capacity or under dry conditions. To make a story short, you can read all this in, well, it will come very soon in Annals of Botany. We made these 3D models, so we used a root architecture to explain that an important factor in the root development is the young roots, well, the emerging new roots for phosphor uptake. You can see that here, if you have the middle phosphor supply, what we found is that dry spells do not reduce phosphor uptake. And that is because you have more new roots that develop. You can see the roots here on this graph. And when they're red, it means that they're taking up more phosphor. And even though the phosphorus is less diffusible, there is more, well, or equal phosphor uptake under dry than under field capacities, because these young roots, emerging roots, go into new zones that are not explored and that bring more phosphor into the system. So the insights that we got from the modeling is that the key drivers in phosphoric uptake are the laterals and the growing root tips. And under dry conditions, we have more growing root tips and more efficient root P acquisition. If you read the papers of Peter Jan de Bouw, that you will also see that there is an interaction with genotypes. So screening for low P tolerance is also screening for, of course, root architecture and their reaction to water. Water supply can be, of course, erratic, can be different, and you can have water stress, but that water stress can be sometimes beneficial for overcoming phosphorus stress because you have more roots and more phosphor uptake. The last point that I'm going to touch upon is our work on new fertilizers. We have been working on developing more efficient fertilizers. And what did we try is we tried to work on the layer double hydroxides. What are layer double hydroxides? These are hydroxides of magnesium hydroxide, in which magnesium is replaced by aluminium, and then you have a positive charge in your hydroxide surface. They are layered, just like clays are layered, but these layers have a net positive charge. So they are anion exchangers. When you make these LDHs, you make them typically from nitrate salts. So they have nitrate in between the layers. You can exchange them for phosphor, and then you have phosphor bound, mostly electrostatic, in between the layers. And the phosphor is trapped, but is reversibly trapped. They can be exchanged for other ions, such as, for example, bicarbonate or carbonate or nitrate or sulfate. They can go in here and remove phosphor from solution. By doing that, you have a slow release fertilizer, but more importantly, you have a fertilizer in which the phosphor is held in a reversible form and is not irreversibly trapped by your soil particles, your iron oxide, or, for example, calcium phosphate. You keep the phosphor in the loop. Our first results were really spectacular. We took an acid soil and we dosed it with a soluble P of phosphor, soluble form of phosphor, and with LDH. We had a massive better growth and a much higher phosphor use efficiency. Hooray, hooray, you could say, then you can go to Nature and publish that. But we started to think, why is this so much more efficient? It's not related or very little related to the slow release properties, but it's related to the fact that in an acid soil, these LDHs are alkaline, make a local alkaline spot, overcome aluminium and manganese toxicity. And when we added gypsum together with that LDH, we had much less difference. So the phosphor use efficiency of also our phosphor, our soluble P was also enhanced. At the lower doses, well, maybe relevant doses, we still had a bit of better with LDH, but in fact, it was not as spectacular. Yeah? And in the calcareous soils, there was hardly any difference. Yeah? What we showed is it works. LDH can be used. It is a slow release fertilizer. And in more recent work with LDH, we have shown that in high pH soils, it gives a beneficial effect on the long term because the phosphor cannot be fixed in calcium phosphate. So it keeps the phosphor more labile, so it can keep the phosphor for a long term. But it is not confirmed yet on the field with long-term trials. It still needs to be done. What we also did with LDH, and that's a nice side story, is that LDH is an anion exchanger that we can use to recycle phosphate. So that's more for the European situation where we have too much phosphor and where we have to recycle phosphor rather than relying on new mineral fertilizers. Urine is is an important source of phosphor, all the urine, animal and human urines, and you can separately collect that. And the phosphate in there is quite readily trapped. You can do different things like struvite, but LDH is also a candidate mineral to recycle P. And you can recycle, in fact, 90% of urine in very short time. It's very quick. You need only 1 kg of that material per cubic meter urine. It's fast, it's binding the phosphate reversible, and it has a good fertilizer value. We also did that now recently. It's in a paper that is submitted on the fertilizer value of urine, raw urine or urine that is recycled through LDHs. I'm looking at my watch and I've been talking far too much. Take home. If we need to capitalize soil fertility in Africa, we need techniques with increased PUE. Otherwise, we have no return on investment. You cannot ask a farmer to invest in 400 kgs phosphor to overcome the phosphor deficiency at once. The key drivers are add organic matter if your soils are also having high iron and a low carbon content. Add the phosphor locally. Check for soil acidity. First overcome soil acidity before you add the phosphor. Genotypes, yes, they're important, but our lesson is that selecting genotypes cannot be done on one location only. G times E interactions are huge. You have to do it at many locations in interaction with organic matter, with pH correction, etc. Otherwise, you have only some ranking that are relevant for the situation. Water management is also important. Of course, you need to add water, or you need, well, the rain is of course essential, but the interaction with phosphor is not all that simple. It's not that drier, it gives you lower phosphor availability. The interaction with genotypes becomes important. There's of course more research needed, and I think one of the things that we have been missing a bit is the testing of the residual value of fertilizer P. So best is that we need to set up some long-term trials in Africa with some what we call rundown plots. So you add, say, phosphor for 10 years and then you stop to identify what the interaction is with genotypes and organic matter. And the genotype work, of course, there's a lot of attention to screening varieties that are more tolerant to low P for whatever reason, rhizosphere reason or root architecture, but you need to do that at different environments. And with that, I would like to stop and I leave the floor open now for questions. So over to you, Margit.
Thank you, Erik, for a very interesting presentation and very clearly presented. And I was looking at the chat box, dialog box, a lot of people, commending you for a very clear presentation. And so thank you very much. So we will move to, so I have in the Q&A box some of the questions that I will bring it up to the screen. And then let me first bring the banners again. All right, and then go to comments. So I will select and project, and you or your colleagues who are online are welcome to respond, as the case may be. So let me go and the list of questions. Okay, I'm on it. Just a second. So I also see there are people connecting from different parts of the world. India, I saw Brazil, Kenya, a lot of people. Okay, Nigeria. So I'm scrolling down my, there's one from New Delhi, International Zinc Association. Let me see. So here is now the, so maybe I'll start, some of those may have been even addressed because they were coming all the way, but this is the one. So Erik, can you see?
Yeah.
Yeah. So I'm curious to know, this is by Abdul Aziz. I'm curious to know if your lab has developed a calibration curve to predict P to apply for corn based on P. Olsen test in the soil.
Answer is yes. For maize, yes, even specific for maize, you can find that back. I think I'm just thinking in the 2018 paper that I cited of Sophie Navarra, these were all the crops. But in the tables, you can go also specifically for the crops, by crops. And for maize, there is another critical value. Well, typically for phosphor, you end up with a P Olsen of between 20 and 30 milligrams of P per kilogram. But mind you, this was done for the source of Europe. We've been looking also to the source of UK specifically because Olsen is a lot used in UK. We have been looking also for the source of Sweden, where we had some data. But the answer is yes, we re-evaluated the Olsen for corn, for maize, but we did not go back to the literature of the Olsen test in the US that were done. But then maybe the question is, what about Africa? And the answer is no. What we definitely need to make calibration line is, in fact, a whole series of fertilizer trials in Africa with associated source in which the source of the different treatments. And that is something that we missed. In fact, in Africa, there has not been a comparative test of soil fertility test for phosphor. We did in large pot trials with large pots, that the one that I tested. I also did one in Madagascar based on flag leaf phosphor, but not on fertilizer responses. But I've been looking around, and yeah, I was wondering, is there an Olsen test calibrated for corn in Africa? The answer is, as far as I know, no.
Okay, thank
And it's
you.
predictable that the values will come out differently. Yeah? You will need higher Olsen P than in the young source of Europe. Yeah.
So here is another one related to soil tests. So please comment on the relative significance of wet chemistry and dry chemistry spectra soil tests for both.
Yeah, good comment. But I'm not very strong in these spectral tests, these spectral source tests. So the NIR and MIR, we have been doing it a few times, but never did that for the source of Africa. But it's predictable that it should work because I mean, with these many of these tests, you determine minerals, and in fact, you could evaluate. You don't measure the phosphorus so well, but you detect the minerals, and you would immediately identify the difference between the high iron and aluminium source and the low iron and aluminium source. But it's all a matter of calibration. As somebody has to do it, and the cost is not the test and the machine. The cost is you need different locations with fertilizer trials, and that's what we need. And that dry test could be, of course, cheaper. You only need a few grams of dried soils to ship to that lab, and they put it in a machine, hit the button, and get an answer. So it's really less than $1 for a test, and it could work, but somebody has to do it. Yeah. Yeah.
So this one is, how did you measure fast pool phosphorus?
Yeah. For the desorption test, the test is what we call the infinite bag. You take a soil sample, put it in suspension, and then you have an adsorbent that you add to the soil suspension that absorbs the phosphor out of the solution. That adsorbent is, not surprising, an iron oxide that is either on a membrane, so we call that could be an iron oxide in a filter paper, could be also an anion exchange membrane. So the last tests were done with anion exchange membranes, or it could be a dialysis bag that has iron oxides. You do that desorption test, so you refresh your iron dialysis bags, or you refresh the membranes. and then you plot the curve, and the curve you can, well, that curve fitting that we do is a 3-parameter curve that has a fast and a slow pool and rate constants. And the fast pool is just a fitting. So the fast pool is determined by everything that with a half-life of desorption between 2 and 5 days, and the slow pool has desorption half-lives of more than 100 days. So it's curve fitting out of that desorption thing.
Okay, so thank you. Next one is there any intercropping system to improve phosphorus use efficiency of organic farming, and how we can calculate the phosphorus use efficiency?
I'm not really the expert on intercropping. Well, we have been doing intercropping, but I'm not really the big expert. Of course, a lot of things is intercropping with a nitrogen fixer, which is then a combination of the nitrogen that you add to it, and that's of course something else. So in all our trials, we have been adding ample nitrogen, but there's also something. There's of course other things as well. The intercrop can often add a lot of organic anions that have a very long persistence in soil. So these things like citrate and oxalate, they are not going away within weeks, and they can stay for a long time and keep the phosphate better in the loop. And then there is it's well known that like a lupin, which has a high citrate exudation, that it can have a good effect on the following crop. So the intercropping with the lupin is always beneficial, or another nitrogen fixing. And then you can have a beneficial effect on the phosphorus use efficiency of the mineral fertilizer that you applied in the crop that in which you are interested.
Okay, so this one is kind of on new technologies, new fertilizer products, nanophosphorus. So fertilizer.
Yeah.
So it's claimed that they reduce the losses and improve availability of P. Is it true, or what do you think of nanophosphorus?
Well, the nanophosphor, we have been trying to make nanofertilizers, and we were not really successful, I must say. I mean, there has been based on apatites, and then we have been basing it on iron oxides that we try to keep in solution, and we never got it really right. So a few people have been trying it and have been successful, but the question is at what price and how long is their long-term effects? Because nano stops to be nano once in the environment. A lot of nanoparticles in solution, when you bring them into a soil, are coagulating and are not nano anymore. There have been successes, but I'm not yet convinced that it is such a large thing. I mean, and definitely not the nano fertilizer that they add to the leaf foliar application of phosphor. You can have some uptake, of course, from the leaves, but it's not the total quantity not the 20 kg can be supplied as foliar. But in fact, maybe other people can get in. I'm not want to say that it's not possible, but what we tried was unsuccessful. Well, or not successful enough to make it into the field. Yeah.
But are there other groups trying to create nanophosphorus? Other labs that you have?
If other labs, I know that in the group of Mike McLaughlin and CFZRO, they have been reasonably successful, but not to bring it into the field, I think, yet. But I mean, well, it's known that colloids are bioavailable. That is work that has been done by one of my postdocs, Daniela Montalvo, who worked in CFZRO. She proved that colloids are bioavailable. We also did tests in solution culture and showed that colloids can migrate in solution towards the roots and so on. And so these either non-natural colloids or synthetic ones that they can move. But don't forget that nanoparticles, they diffuse slower than phosphate. They diffuse up to 100 times slower towards the root. So they are a bit slower in moving. It's of course a lot of phosphor together, but it moving slower towards the roots.
Okay, got it. So this one is, why can't I read it? Just a minute. Show. Oh, it's frozen. Okay. Okay. Can you read
Yeah,
this? I'm trying to. Yeah.
aging, fixation.
Yeah. Easy. Yeah.
Well, what we recently did with that slow release, the LDH, we added it to a soil of that has free calcium carbonate. The fertilizer value was equivalent to that of TSP, or was it DAP? I can't remember. So it was not that say there was no added benefit. But when we analyzed the soil after the plant growth, we found more labile P in the soil. It was detected with isotope exchange than in the soils in which you added as soluble P. So the slow release, I mean, you keep that into and between the layers, the phosphate. That's the idea. So it cannot be trapped as calcium phosphate and fixed as such irreversible. So it stays in the loop. But that was done in pot trials and not yet in the big field things. So the LDHs, also for the people of the fertilizer industry, we can make LDHs with 7% P. So that's something like 15-16% P2O5. It's still not is not yet a fertilizer with 40% of P2O5. It's still a lot of magnesium aluminium that is into it. Yeah. And that these are all things to take into account. Yeah.
Yeah. Okay. So is it the LDH scalable? Like if we,
I think I've seen one fertilizer company trying it in the field yet. Yes, yeah, at large level, yeah, it is scalable. Yeah, and
okay.
we are continuing on that avenue. So the LDH is not stopped. There's all the work that will start soon on LDH. Yeah, but yeah, it's not yet. I mean, it's not yet widely used. That thing of LDH for phosphor recycling, it's an alternative. Struvite in Europe, they all say struvite, but struvite also is expensive to make. Well, expensive. It's more expensive than mineral fertilizer. So the LDH it can be a competitor also for that phosphor recycling. That's the one thing that we're trying. Well, huh? Yeah.
Yeah. So this is a question which has come a couple of times. So I'm combining it. I won't bring the other one. So the basic idea is, okay, you have the phosphorus inorganic fertilizer as an input, and then the phosphate solubilization, the bacteria that do, or, microbes.
Yeah.
So that's a challenge. And is it effectively tried in the African context? I mean, combination of these phosphorus solubilizer microbes with going with the fertilizer, inorganic fertilizer.
Well, the interaction with the microbes, I cannot answer too much, but what is also clear is the whole work on rock phosphate. Can rock phosphate be used as such rather than the acidulated phosphor? In some conditions, you do find a large success of rock phosphate in interaction with selected crops that in fact the rock phosphate, if it's finely ground, gives better results than just the acidylated phosphor. But in terms of identifying microbes that are able to dissolve the phosphor that is inherently in the soil, well, it is possible, but the question is it agricultural? um can it be used in agriculture and with a good return on investment? I'm always a bit skeptical on success stories of microbes. I mean, microbes in itself, there's very little nutrients that are in the microbes themselves, and that microbes can unlock phosphor from the iron minerals. Yeah, that is quite difficult. And question is it sufficient? It can do a little bit, but just an inoculation with microbes to access the phosphor that is fixed in the natural source, I'm very, very skeptical about that. That is, it's not like nitrogen and which of course it can be mineralized, but to unlock phosphor. I mean, try also in the lab to mine phosphor. Many of these soils have, when you just make an extract, have no detectable phosphor with the classic methods. I mean, with the molybdate blue or malachite green methods, you hardly see. So let be that you can open up the structure. You can try to, for example, make it anaerobic to reduce the iron 3 to iron 2, but when you have a lot of crystalline iron, that is going to be very, very difficult. Yeah.
Okay.
Yeah.
Yeah, thank you. So here is another one. What is your Victor Gustavo asking? What is your opinion about anionic polymers applied
Yeah.
in pea fertilizers to improve phosphorus dynamics in highly weathered soils?
I have very little experience, but it's not a bad idea to do that. It is definitely it's a good idea to work on that avenue as well, to increase fossil use efficiency, to have fertilizers either coated or associated with anionic polymers that locally block binding sites. That is, yeah, I'm not an expert. Well, we did a little bit of work on anionic polymers and surfactants that we added, but in these, in the combination with phosphor and highly weathered soils, I have not done. But in terms of chemistry, yes, that's a good idea. Yeah. But of course, the question will be then still, I mean, is it economically viable? But it can, yeah, it can. If sometimes it's a matter of just that little tipping point to make it economically viable for Africa. Yeah.
Yeah, got it. Thank you. This one is from Morocco.
Yeah.
Mohammed asking, in many cases in Morocco, we base fertilizer recommendations in soil analysis, based on soil analysis. How can we figure out the aging part of it?
Yep. You need aging. You need time.
Hmm.
Until you lose all your hair, just like me, and that's aging. Um, you need, well, there is a need for long-term fertilizer trials, which, I mean, there have been a few attempts, and I know one in Nigeria that has been in trying, but in a good way of doing it is you do set up your trials for 20, 30 years, and then you do rundown trials. So you have the sustained supply and then the rundown trials, and then you do your crop response, and then you do your soil tests. Yeah? Now, as a chemist, you would say, well, you don't need that test. Just do, say, test a DGT and measure soluble P, and then all the aging is taken into account. That's true, but that's from a conceptual point of view. The truth of the pudding is in the eating. So you better, I mean, there's many good reasons to set up long-term trials with phosphor in these highly weathered soils to identify what happens on the long term.
Okay, this is more like a comment from Abraham. In Nigeria, fertilizers are highly politicized, making it more expensive than they should be. Again, pea fertilizers are not usually found as straight fertilizers. So this case is typical in Southeast Nigeria.
Yeah, I guess comments is referring to the fact that they are all blended fertilizer NPK things. Um, yeah, but you need the other nutrients as well. And of course, it depends on the local situation in Nigeria. Remember that we've seen quite some sulfur deficiency and magnesium also, magnesium and sulfur. Um, and of course, yeah, the NPK blends should be valuable for the typical situation. I cannot judge from here which blend is which NPK blend is the best for that. But of course, the price, yeah, that's a classic thing. We all realize that the whole economy behind is a major factor. The availability and the price of the fertilizers, but well, you I don't have to convince most of you is that we need that. You need to have a good distribution of the fertilizers and a whole distribution of it, and then you can have a return of investment of definitely well above one, and then you capitalize your soil fertility, and after many years you will see that you can reduce your phosphor needs, and then you can reduce the cost, of course, and then the whole agriculture will become much more efficient. Yeah.
Thank you. This one is Obey. Before you conclude, please go over the definition, determination of PS1. PSI?
Yeah, the PSI. That is, it's something that we use in the lab. Often, it is what you do is take a soil, take an extract of the soil, and you distribute over the different tubes and different replicas, and then you titrate your soil with increasing doses of phosphor, a known quantity that you add to each of the suspension, and then you shake them for 24 hours, then centrifuge the suspension, measure the phosphor in the solution, and then you determine at which point you obtain 0.2 milligrams of phosphor in solution. And in that tube, in which you reach that, and then you go back to your paper, how much phosphor did I add to that tube? And that's your phosphor saturation index.
Yeah. So, Erik, I'm going through the chat box, a lot of comments on how nicely you presented and the insights, very useful. So, I'm not reading that, but they are. I think these webinars will be archived, and probably we can access the conversation that happened. So this is the one. It says the presentation was so impressive. There are lots of decision support tools, so which are listed here: DSAT and SWAT and QUEFTS. How can we address the usability of these tools in relation to P recommendations?
Well, I'm not the person to talk to, but Peter Pepers, who comes from our group and who works at IITA in Kenya, has also included field trial responses of phosphor and decision support systems. So he is the person, he gave a presentation at the phosphor conference in 2018 in Leuven about that decision support system, which includes also phosphor. If I recall it right, it has soil information from the soil map that has the indication of to what extent it is likely that you have phosphor deficiency. So that likelihood is related to, of course, the weathering stage, the type of soils, and which is in soil information databases. So you can have, when you have your GPS coordinates from which you are, and you have that app, these systems, they can tell you whether you have a likelihood of phosphor deficiency. Of course, it has not the site-specific information of a soil test. It's not using a soil test, but it's just using the soil information. And so there is already information on that. So Peter Pepers, Pypers, is a person who you have to Google and ask about this.
Great, thank you. And here is another one comment. Okay, what is your comment on P reserve depletion, future of food supply, and measures to put in place? Yeah, we are all dealing with a finite resource.
Yeah, we are dealing with a finite resource, but at the moment, in the near future, it will not be depleted. I mean, that's well, you know, the peak P of 2000 and was it 2010 or 2008? After that peak, the phosphor price, then it decreased again, showing that it's when they open new mines or whatever, increase the production a bit. But of course, on the long term, it's going to be depleted. But I think that at the moment, for capitalizing phosphor fertility in Africa, the main focus should be on finding the ways to enhance the phosphor use efficiency. We cannot afford of adding 2 tons of phosphor per hectare in all the fields. That's impossible. Maybe they need in the end 2 tons, but the way for development is the way of using the little bits as efficient as possible, which can be with all, kind of approaches that I explained. Yeah. So the combination of all these techniques.
Yeah, great. So, Amari asking, hello, great lecture. Thank you. One question: to what extent a P fertilizer coated with humic material could help?
It has been tried, but it's same like the anionic polymers. I mean, humic substance is an anionic polymer. I'm not the expert on that, how good that works, but it's definitely a way to identify that. So the way we did it was the cheap way. We bought the TSP on the market, the locally available TSP, and we mixed it with the locally available organic matter. The compost we call the farmyard manure, but it's mainly that organic matter. Don't forget that a large deal of our effects were related to the pH effect, which you will not have when you coat that with humic substances. So just adding the organic matter is definitely more than only blocking binding sites. It's also that pH effect, which is large when you have a low pH. And it's of course not for the entire of Africa. It's not because we have found it in many places that it's the entire of Africa. So acid source, you can also have pH neutral source in Africa, and calcareous source as well.
So we are coming close to our time, and
Yep.
I might take just a couple more, and then if any remains unanswered, we can do offline. I do appreciate Erik's very generous with his time with us today. So this question from Zamima. PUE was 10% in highly weathered soil. It was possible to increase it to 15% with farmyard manure. What about the fate of the 85% fixed P? How much can these be desorbed with time and P addition or other strategies?
Yeah, the 85 is not lost. So to make it to explain that, so what you see we did several field trials for each 3 or 4 years, and you see that the phosphor use efficiency increases at the same locations. That is because you have the residual value of the one that is before as well. So it's not lost, 100% lost. So the 85, of course, the 85 has lower phosphor use efficiency than the freshly added. How much can these desorb? That is, as I said, this is something that we have not looked too much into it. So we know that the residual value is lower than fresh, but we don't know how much lower. And that is the reason why I advocate long-term trials that include rundown trials, so that you do add phosphor and then you, of course, then you leave it. And what you lose, of course, is the benefit of the placement. So you have added it together with a seed in year 1, the year 2. I mean, you can never add the same seedling on exactly the same spot. So you do that, you lose that the benefit of the placement. So that is something you have to take into account.
Thanks. So this is another one in the context of slightly alkaline soils. To ensure P availability, when should the P enriched compost be brought to the maize crop?
Oh, well, I'm not sure about that question. I mean, because then, well, the organic matter, I would say, has less effect in a slightly alkaline soil. There is not that large beneficial effect of organic matter beyond its other effects. Of organic matter always have beneficial effect for many other reasons. Water holding capacity, micronutrients, etc., and the nitrogen and so on. But I'm not sure if there would be a beneficial effect like for keeping the phosphor better in the loop and avoiding phosphor fixation. So I'm not sure about that.
Yeah. So this one from Brazil, Luis Roberto. Could you tell us something about your experience with Malik 1 as an indicator of P availability in Africa, as we do in Brazil?
Hello, Bebeto. Yeah, I know Bebeto.
Uh,
Everybody knows Bebeto. No, honestly, no experience with melee. We have not done in this source. We did not include melee, and I know it works. If you say Bebeto that it works well in Brazil, then yeah. Well, in Brazil you also use that anion exchange membrane that has been developed as well. But no, I have no experience with melee in African soils.
a short one. Did you apply farmyard manure annually to maintain the soil organic matter?
Yes, answer is yes, but we added it. It was not to keep the soil organic matter up to standards, but it was just in the planting hole. So we did not broadcast farmyard manure. So in all the trials that we did, we added in the planting holes, except for the paddy rice trials. That was different. But so on purpose, we added farmyard manure in the planting hole. It's of course it needs more labor. Everything was hand put by hand in the soils, not with machines. But it was in all the experiments. Yes, it was annually. It was not a one dose, but annual dose.
So I think with that, we'll conclude the Q&A session. Thanks a lot, Erik, for your time and exchange of knowledge, ideas, experiences in Africa, techniques for peak chemistry. Really, really very informative. And also equally, I thank all the participants, who asked the questions and for a very informative discussion. That has happened. And if any we have missed questions, please feel free to either email me and offline we can address that. And one thing I want to encourage is that one of the purpose of bringing such authorities in the field is to also make collaboration linkages, whether at the teaching level or research collaboration levels or OCP, as a company. So, Erik, there have been a lot of people connecting from different parts who are in your audience. So it's good. And so I'm asking them to, explore potential ways of working together, and especially your passion and commitment for Africa. So that would be great. And in the end, I want to show you our next webinar, which will be same time next Monday. And that will be, let me bring up the banner for that. Where is that? Okay. Here it is. Do you see that?
Yes.
So this will be a webinar by Professor Manish Raizada. He's a full professor at the University of Guelph in Canada. And he will speak on opportunities for agri-science collaboration within Africa. So in the Agriculture Scientists for Africa group, we are trying also to identify what are the main institutions within Africa for the knowledge of people in, scientists or students working in North America or Europe. So Manish has come out with a list of key partners that he will bring in. And we thought that maybe also promote further collaboration. And with that, we are at the end of our webinar. Erik, any closing comments or last message?
Oh, I just want to thank Mieke and Pieter Jan. I think they're still in the audience to help me putting this presentation together. It's definitely not a one-man show. Well, I gave a one-man show. Unfortunately, they did not join the question part, but I want to thank them.
I tried to bring them on the screen. Yeah, thank you, Mike and Peter Jan. Yeah.
Mieke. You're welcome, Erik.
Okay, Mieke, we can hear her. And Pieter Jan, I don't know if we can hear him.
Yes, thanks.
Well okay, done. Yeah. Okay.
Thank you so much for joining, and very interesting and applied work you are doing. Best wishes. Yeah.
Yeah.
All the best.
Okay.
Thank you, Erik, again, once again.
Have a good time.
It was a real pleasure and very productive.
Okay. Stay safe. Yeah. Goodbye, everybody.
Goodbye, and we'll end the broadcast now.
Yeah.